Hello, I'm Tim Sandle and I'm Head of QA GXP Compliance and Sterility Assurance. Welcome to the 2024 Sterility Assurance Refresher Training. And for this year, we've decided to focus on the changes to an aspect of GMP that governs everything we do, which is Annex 1 to the Orange Guide. And this annex was updated last year and we've been putting it into practice. So what we're going to do in this session is focus on the key changes. Okay, to begin with, we're going to have a look at what is Annex 1. So Annex 1 covers everything we need to know for the manufacture of sterile medicinal products. It forms part of the GMP regulation that governs the European Union, many countries around the world, but importantly in this context the UK as part of the Orange Guide, and it's legally enforceable as part of the 2021 UK Medicines Act. It had a substantial update in 2022, with the new version coming into force in 2023. And our sterility assurance processes and our approaches to contamination control have been updated to meet the new Annex 1 requirements. So if we look at the new annex and the old annex, the first thing that jumps to mind is that it's much bigger. It's gone from 15 to 50 pages. And there's also hundreds of more clauses. But beyond the size and weight of the new document, it's more science-based. It's more risk-based. And it captures some of the advances and changes with sterile manufacturing technology that have taken place over the past decade. In particular is an emphasis on the importance of barrier technology. So wherever possible we should separate people from product, and we should always minimize and seek to eliminate the presence of people within grade A environments, particularly where product is dispensed and stoppered. And it also allows us to embrace new technologies and new techniques. Fundamental to many things within the annex is a requirement to use quality risk management. And these are proactive tools that need to be followed and formally written up to think about where contamination could occur, and then to find measures to reduce and mitigate the contamination, and then finally to try and monitor to check that our contamination system is working properly. And with every risk assessment we need to keep asking ourselves, what are we doing to protect the product quality and to guarantee patient safety? Part 2, the contamination control strategy. So another key thing within the annex is a requirement for every facility to have a contamination control strategy. A contamination control strategy needs to be scientifically established. It needs to be based on quality risk management, which we were just looking at. It defines controls. It's maintained and it captures knowledge. So all of the best practices, all of the good ideas, all of the continuous improvement practices that we come up with should be captured with updates to the contamination control strategy. It also needs to be holistic, which is something that we'll come on to shortly. But the contamination control strategy will go down to the level of how often should we change gauntlets on an isolator? How often should we clean and disinfect a clean room? What disinfectant should we use? And so on. So it's answering all of those key points for maintaining contamination control. It also looks at contamination in a wider context. So there are microorganisms, bacteria and fungi, and it makes references to our viral pathogen safety procedures as well. It also looks at microbial byproducts which can be released as microorganisms are living or when they die. So, for example, endotoxin. It also looks at other sources of pyrogens. So if pyrogens are present in, say, parental drug products, they can initiate fever in the patient who receives it. There's also particles, both particles that are visible and those that are non-visible. So non-visible particles can interfere with the way the medicine works in the body. Visible particulates are impurities like, say, glass fragments. And then there's also chemicals as well. So things like cleaning, validation, and also any residues from our cleaning materials need to be captured within the strategy. Part 3. Let's now have a look at some of the key changes to the Annex in detail. So some of the key changes in the Annex. So first of all, we have a section on people, and people are of such great importance. We cannot manufacture our sterile medicines without people. And what the Annex is doing here, it says we should always have the appropriate numbers of people. So this means, say for example, if we're running a filling line and it says 3 people, it says 3 people in our procedures for a reason. That's the number we need to carefully complete an aseptic operation. We also need highly trained people. So we need people with appropriate knowledge, skills and experience, and that means a basic understanding of sterility assurance and microbiology. We also need to control the numbers of people within a clean room. Every additional person who goes into a clean room increases the particle concentration by around 50,000 particles. So we always want to have as fewer people in a clean room as possible. And then we have good behaviours and appropriate aseptic practices, and that's particularly important when we're interacting with the grade A zone, such as within a filling machine. There are also changes within clean rooms. So I've selected a few of the changes here. So the first one is that there's a requirement to assess, measure airflow velocity under unidirectional airflow devices across the entire range of the device. So this is generally something we'd address in validation, but it can affect the points that we might use for routine checks. There's also a requirement to perform more detailed airflow studies, particularly where grade C meets grade B. So transfer hatches, airlocks, opening autoclaves, decontamination chambers and so on. And this is to support the other airflow studies. And of course, airflow studies are what's sometimes referred to as smoke studies. There's also a requirement to assess the recovery rate for a clean room. So what this means, if there's a particle event that takes the number of particles above the limit within the clean room, how quickly can the clean room recover? And the Annex requires that to be within 15 to 20 minutes. There's also a lot more emphasis on data trend analysis, understanding how the clean room performs both at rest, so that's when there's no people in it, and in operation, that means when we're in it and the machines are running. And there's also a requirement to perform risk assessments to state why the environmental monitoring samples are in the particular locations they are. So what are we actually trying to measure, and what does an excursion actually mean from a given location? There's also a strong emphasis upon segregation and mix up. So first of all, different activities should take place within different clean rooms. So we would have a dedicated area for the preparation of sterile components. So this is why sterile supplies is in a demarcated area. There also needs to be procedural controls, as in SOPs, process instructions and so on, that are designed to prevent mix-up or cross-contamination. There also needs to be a lot of detail on design of clean rooms and equipment and facilities. So this diagram here might look a little bit complicated, but it's not intended to be. It's basically about the different layers that surround the most important aspects. So red on the slide there is grade A. So grade A is protected by a barrier, but it's also protected by the grade B clean room. And then the grade B clean room, which is the darker blue, is protected by the lighter blue, which is the grade C. And then the grade C is protected by the grey colour, which is the grade D. And then finally in the white zone would be a CNC, a controlled not classified area. A good way to think about this is a bit like the layers as you unpeel an onion. You're protecting that core with a contamination control cascade. Also in Annex 1 there are a number of changes to utilities. So first of all, there are things called critical utilities, and these are things that could come into direct contact with the product. So pharmaceutical grade water, we use water for injections to formulate our product. Gases, compressed air and nitrogen, and also the steam that provides the clean steam to an autoclave. Furthermore, with utilities, there's a lot around water controls, so how the water system is designed and maintained. So things like the orientation of the pipework, the speed at which water flows through the pipework is really important, because fast turbulent flow of water stops any microorganisms from adhering to pipework. When microorganisms get stuck to pipework, that gives them an opportunity to form biofilms. And the greater controls are around the water system, the water injection system, and that's required to be maintained at a temperature above 70 degrees Celsius, because again a hot water system creates conditions that are not favourable for microbial growth. Part 4. Now we're going to have a look at what Annex 1 is saying about the holistic approach to contamination control. So again, this is another illustration of what we're trying to achieve. And again, it's about this holistic approach and this cascade of contamination. So again, we have the red area, which is grade A. And you can see there's a reference below that to less than 1 CFU. So that means that we're targeting within grade A no microorganisms whatsoever. Moving up, so we're going leftwards across the diagram, we have grade B. So there's a reference there to lots of number 5s and a number 10. So the 5s are reference to the surface sample limits and the finger plate limits, which is no more than 5 colony forming units, and the reference to 10 is for air. Jumping further backwards, we get to grade C, and here we have much higher permitted limits. We're allowed 50 microorganisms on a settle plate, 25 microorganisms from a surface, and then in grade D we're up into the hundreds. So we can see there we have different levels of permitted microorganisms, and as we move back to grade A, we're seeking tighter contamination control. We won't go into detail now, but above that are particulate limits. If we just turn our attention to the arrow at the bottom, then this is the pressure cascade. So this is another area of protection. We want the air throughout the facility to be flowing from grade A to grade B, from grade B to grade C, from grade C to grade D, and grade D out. So we are seeking to make it harder for airborne microorganisms to progress into our most important areas. And again, it's all about protecting grade A. Transfer controls. So transfer controls are particularly focused on how items will get into an aseptic filling suite. So there's strong protections around grade C to grade B, and then from grade B to grade A. So the strongest measures are with deparogenation and sterilisation. For items that don't go through a deparogenation tunnel or an autoclave, then these would be single-use sterile disposable items that would ideally go through a decontamination chamber with an automated VHP cycle, so decontamination chamber. And there are provisions to pass some items through transfer hatches, which would be protected with a localized unidirectional airflow, but which would require manual disinfection. And it's very important that that manual disinfection is undertaken using a sporicidal disinfection agent. I mentioned earlier that one of the key things about Annex 1 that it's trying to get across in this new edition is the importance of barrier controls. So barrier controls essentially either means a RABS. So RABS stands for Restricted Access Barrier System, so lines 1 and 2, or an isolator. Isolators are superior to RABS because they're fully enclosed. They have an automated VHP decontamination cycle, and access is only permitted through gauntlets. A RABS is not completely enclosed and can only be manually disinfected. But a RABS is still a very strong and safe system, but we just need to be more mindful about personnel controls and the way that we interact with the RABS for setup, or if there was a need for any open door intervention. There's a lot more emphasis upon aseptic behaviours and aseptic controls. Another thing with isolators is that isolators are very good, but they're not completely infallible. So positioning items both in an isolator and say a decontamination chamber for VHP is really important. We cannot have any two items touching, and items need to be positioned and presented so that there are no folds in the packaging. So what we're always trying to do is avoid what we will call occluded surfaces. The reason for that is that it's an area where the VHP won't reach. If the VHP won't reach, then microorganisms could survive. Another thing with isolators is that we need to be very careful when we use gloves and gauntlets, because there is a degree of fragility and they can sometimes become broken, torn, and this then could compromise the status of the isolator and lead to a contamination event. Part 5, sterility and asepsis. So there are an array of sterilisation technologies that are addressed within the new Annex. The preferred and most robust method of sterilisation is heat. So this can either be moist heat, which uses superheated steam, as in the case of an autoclave, or it can be dry heat, which essentially decontaminates through infrared technology, and that would be, an example would be a depyrogenation tunnel. There are a number of items that come as single-use disposable technologies, and these can be sterilised using other types of sterilisation technology, and these need to be assessed for their suitability. So we have gamma radiation, we have electron beam, which is showers of electrons, X-rays. Within the facility, another form of sterilisation that's very important is sterile filtration. So this is where liquid products pass through a 0.22 micron filter, which allows product through and stops bacteria and fungi from getting through. They're just slightly larger than the pore size of the filter. But we need to assess that the filter was good before use, after it's been sterilised, so the infamous pup sit, and then after use as well. So integrity testing is a really key element for filters. Hydrogen peroxide vapour is a very efficient way of decontaminating, but it is not a sterilisation technology. The reason for that is that it only treats the outside of a surface. Hydrogen peroxide vapour gas is unable to penetrate through packaging. That's why there's additional controls around VHP. There are some other sterilisation aspects which are emphasised in the new Annex. I'll just select some of these. The first one is that sterile filtration through the 0.22 micron filter should be carried out as close to the point of filling as possible. I've mentioned integrity testing. It's also important that all filters are validated. Now, validating a filter involves testing it with the actual product and using a really high bacterial challenge of a very tiny bacterium. So the very high bacterial challenge is 10 million, and if you're interested in the bacterium, it's one called Brevondomonas diminuta. We also want to make sure that filters don't add impurities into the product. So filters would be checked for leachables and extractables, and also we want to make sure that we're not going to get too much absorption, otherwise we could end up with a loss of yield. Single-use sterile disposable technologies have made things much better. Aseptic connectors, aseptic quick connectors is an example of that, and there have been a number of advances, and this is an area that will continue to expand. Media fills, so media fills have been given the slightly posher title of aseptic process simulations, or APS, but media fills again are given a lot of emphasis in the new Annex. And very quickly, some of the key criteria for media fills are that they must capture the whole time of a product that's been filtered. So that's why media would go into an aseptic filling suite, and it needs to be held for the maximum time that product can be held for. The broth has got to be suitable. Every aspect of the aseptic filling activity needs to be captured. So that includes idle process time. So that's why a pre-fill time, for example, would need to be captured. All of the manipulations and interventions that might occur during a normal filling operation need to be captured. The number of vials filled needs to be representative and at least big enough to capture things like shift changes and all of the manipulations and interventions. There should also be a proper incubation, so media fill vials are incubated for 14 days at 2 temperatures. They're inspected and then the media is tested afterwards to show that it would actually support any microorganisms that might be there. And all media fills have an acceptance criteria, a specification of no growth. So one single vial showing turbidity would be a failure of that media fill. Because of the limitations of VHP, because it's not a penetrating technology, we have to independently sterilize product contact parts. And this is both direct product contact parts and indirect product contact parts. Of particular importance are filling needles, stopper bowls, and the guides that the stoppers would move down. And that applies to both RABs and isolators. Sterilisation doesn't stop when a product's gone to inspection, labelling and packaging. We need to guarantee that the product remains sterile until it reaches the hands of the clinician or the patient. So GDP, good distribution practice, includes container integrity, and that means that there's no deterioration of the stopper oversealed into the top of the container, that there's no chance of microorganisms ingressing in, and that remains integral even if we send a product to Australia and it's subjected to the extreme pressures and low temperatures, say you might find in the hold of an aircraft. Part 6, cleaning, disinfection and environmental monitoring. Cleaning and disinfection is given a lot of additional attention in the Annex. There's particularly a strong emphasis upon the importance of cleaning before disinfection. So if we don't clean effectively, disinfectants don't work very well, because disinfectants are not very good at penetrating any kind of soil, grease, protein that might be on top of the microorganism. So they need that to be removed so the disinfectant can do its job. Disinfectants in aseptic filling areas need to be sterile. We also need to rotate so we can increase the spectrum of activity, and this helps guard against the nastier microorganisms that we might find. And we have to use a sporicide regularly, and the sporicide is the thing that's most effective against fungi and of course bacterial spores. Environmental monitoring should always be targeted in the operational dynamic state when there's people around. There are, as well as like standard activities, batch related activities, one of the strengths of environmental monitoring is kind of targeting special events, so maintenance work, coming out of shutdown, checking how well cleaning and disinfection is working when we get changes to room occupancy. Another thing that's important about environmental monitoring is not only that there might be an alert and action level to signal the status of the clean room, but there's also the ability to identify the microorganisms, and that gives a way to look for some of these more objectionable, nasty, strange organisms that signal a breakdown. So for example, if we find fungi in aseptic filling area, that normally indicates that there's been some form of control breakdown somewhere or other, and we need to start forensically trying to find out where that contamination came from. Part 7. Elstrie's own contamination control strategy. So just for awareness, I've mentioned contamination control strategy. We have our own contamination control strategy. There's a screenshot of it. It's called BPL 00001 CSD. It's available in control documents if you're interested in reading it. It's a 60-odd page document, and it sets out our approach to contamination control, and it's a brilliant read. The structure of the contamination control strategy is kind of divided into different pillars. So there's things looking at processes, equipment and people, environment, cleanroom design and the approach to cleaning and disinfection, design of utilities, approaches to improving aseptic processing, so single-use technology, barrier technology, sterilisation, and then some wider quality control aspects like vendor approval, release of materials, monitoring strategy, and so on. So it kind of captures the things that I've been discussing. So you can see the importance of cleanroom design, the surrounding importance of utilities, applying scientific knowledge, experience and risk management, and all of these elements come together, and if we focus our eyes on the red, the grade A, that's what we're seeking to do all we can to stop product becoming contaminated all the way through, but particularly in the grade A zone. Part 8, let's wrap up. Just carrying out monitoring does not ensure sterility. So we need a contamination control strategy. We need to identify hazards, find ways of eliminating them, mitigate risks, apply risk management to do this, and then monitor to prove that everything is working. We need good design of premises, equipment and processes. We need highly qualified and skilled people like yourselves. We need good designed procedures, and we need to put it all together. We need a joined up approach. We need to be able to stand back and look at the big picture. So it's this holistic approach to the assurance of sterility. Thank you very much for watching, and thank you very much for taking part in the 2024 Sterility Assurance Refresher. I hope it's been enjoyable, and hopefully you've gained a flavour of what the revised Annex 1 is trying to say. So I'm Tim Sandle, Head of QA GXP Compliance and Sterility Assurance. Thank you for watching. Have a great day.